Hello everyone, my name is Raul Urquiola Perez Chacon, and my project is titled Antibacterial Effects of cCopaiba oil and T4 Bacteriophage against wild-type and Ampicillin-Resistant Escherichia coli. Antibiotic resistance in E. coli is a growing public health concern because it reduces the effectiveness of conventional antimicrobial treatments. Bacteriophages are viruses that infect and lyse bacteria, so they're being studied as possible alternatives or complements to antibiotics. cCopaiba oil is a natural product from Copaifera species that has been reported to have antimicrobial properties. However, there is still limited research on the combined effects of cCopaiba oil and bacteriophages against E. coli. On that basis, the study investigated the antibacterial effects of cCopaiba oil and T4 bacteriophage against wild-type E. coli and also examined the effects of combining cCopaiba oil, T4 bacteriophage, and ampicillin against an ampicillin-resistant E. coli strain. My hypothesis was that cCopaiba oil alone or in combination with T4 bacteriophage would reduce E. coli growth. For the methods, wild-type E. coli was cultured in nutrient broth and nutrient agar at 37 degrees Celsius. I also generated an ampicillin-resistant E. coli strain through a stepwise exposure to increasing ampicillin concentrations from 1 to 100 micrograms per milliliter. Resistance induction was confirmed by growth in both broth and agar, and representative cultures were examined by gram staining. T4 bacteriophage was propagated using an adapted plaque overlay assay based on the phage on tap method, and phage titers were calculated in PFU per milliliter. cCopaiba oil emulsions were prepared from 1 to 100% using 1% twin 80. Antibacterial activity was evaluated using minimum inhibitory concentration (MIC) and zone of inhibition (ZOI) assays against wild-type E. coli because the absorbance at OD60 based MIC results were inconclusive. Selected treatments were further analyzed using a most probable number or MPN assay. Finally, combination plaque overlay assays were performed for wild-type E. coli with T4 bacteriophage and cCopaiba oil, and for the ampicillin-resistant E. coli with T4 bacteriophage, cCopaiba oil, and ampicillin. Moving into the results, the first major finding was that T4 bacteriophage was successfully propagated across 5 successive plaque assays. The lysates used in downstream experiments range from 1.02 times 10 to the 4th to 6.5 times 10 to the 4th PFU per milliliter. In figure 1, you can see a representative plaque assay plate from the 5th propagation. That plate corresponds to the 1 to 100 dilution and contained 389 plaques, confirming successful phage replication and lactate activity. The second major finding was successful induction of antibiotic resistance. Stepwise exposure produced E. coli capable of growth at 100 micrograms per milliliter ampicillin. Growth was observed across the full concentration range tested, and the gram stain in figure 2 shows red rod-shaped bacteria, which is consistent with the gram-negative bacilli and supports that the strain remained morphologically consistent with E. coli. The third major finding involved cCopaiba oil activity against wild-type E. coli. In the absorbance from the MIC assay, it increased across all treatments over 48 hours, so no MIC was obtained. In this ZOI assay shown in figure 3, only the 100% cCopaiba oil treatment produced measurable inhibition, with a mean inhibition zone width of 2.47 plus or minus 0.06 milliliters. So at this stage, cCopaiba oil appeared to have only limited antibacterial activity when measured using standard MIC and ZOI methods. However, the most important result of my project came from the MPN follow-up, shown in figure 4. Because the MIC results were inconclusive, I used MPN to estimate the viable bacterial abundance in selected treatments. This revealed a strong decrease in viable bacteria with increasing cCopaiba oil concentration, and the 100% treatment fell below detection. All cCopaiba oil treatments differed significantly from the negative control, and the 100% treatment also differs significantly from the 80, 40, and 20% treatments. So even though the absorbance readings suggested continued growth, the viable count methods showed that higher cCopaiba oil concentrations were actually reducing bacterial survival. This was one of the most interesting findings in the project because it showed that the MPN assay captured an antibacterial effect that the plate reader did not. Lastly, I looked at the combination treatments. These plaque overlay assays produced variable results. In the wild-type background, higher oil concentrations altered plate appearance, but plaque identity was sometimes uncertain. In the ampicillin-resistant background, shown in Figure 5, the clearest plaque formation was observed at 80% cCopaiba oil. Overall, these combination effects were suggestive but inconsistent, so they did not show the strong enhancement I originally expected. In conclusion, cCopaiba oil did show antibacterial effects against wild-type E. coli, but these effects were much more clearly detected by MPN than by MIC or cCopaiba assays. T4 bacteriophage was successfully propagated and remained effective throughout the study. I also successfully generated an ampicillin-resistant E. coli strain capable of growth at 100 micrograms per milliliter ampicillin. The combination treatments show some altered growth patterns, but their effects were variable and not consistently stronger than the individual treatments. Overall, I think the most important findings of this work were the successful induction of antibiotic resistance, the use of MPN to reveal antibacterial effects not captured by absorbance, and the suggestive but inconsistent interaction between cCopaiba oil and T4 bacteriophage. Future work should use higher phage titers, more standardized cCopaiba oil preparations, more biological replicates, and quantitative follow-up of plaque-positive treatments. Thank you.